Now let's talk about some other kinds of cells that we would find in the nervous system, specifically glial cells. Glial cells, sometimes referred to as neuroglia, occur within both the central nervous system and peripheral nervous system. Glial cells differ from neurons in that they are smaller and capable of mitosis. Glial cells do not transmit nerve impulses, but they do assist neurons with their functions. Collectively, the glial cells physically protect and help nourish neurons and provide an organized, supporting framework for all the nervous tissue. During development, glial cells form the framework that guides young migrating neurons to their destinations. Glial cells outnumber neurons. The nervous tissue of a young adult may contain 35 to 100 billion neurons and 100 billion to 1 trillion glial cells. Collectively, glial cells account for roughly half the volume of the nervous system. Even though the numbers of the glial cells far outnumber neuronal cells are still considered to be the functional unit of the nervous system. The specific kinds of glial cells that we find in the central nervous system are astrocytes, ependymal cells, microglial cells, and the oligodendrocytes. Let's talk about astrocytes first. Astrocytes exhibit a star-like shape due to the many projections from their surface. These numerous cell processes touch both capillary walls and different parts of neurons. Astrocytes are the most abundant glial cell in the central nervous system, and they constitute over 90% of the nervous tissue in some areas of the brain. Their functions include helping to form the blood-brain barrier. They help regulate tissue fluid composition. They can help regulate synaptic transmission. They form a structural network. They help with replacing damaged neurons, and they assist in neuronal development. As far as regulating synaptic transmission, some astrocytes help remove neurotransmitter from a synaptic cleft. As I like to tell my physiology students, a message that is sent is only effective if you send it when you mean it, and you don't keep sending it when you don't mean it. So the neurotransmitter that's released by a neuron should only have a short half-life in the synaptic cleft. The signal should be respond when there is no longer an action necessary. What about microglia? Microglial cells represent the smallest percentage of CNS cells. Some estimates of their prevalence are as low as 5%. Microglial cells are typically small cells that have slender branches extending from the main cell body. They wander through the central nervous system and replicate in response to an infection. They perform phagocytic activity and remove debris from dead or damaged nervous tissue. Thus, the activities of microglial cells resemble those of the macrophages of the immune systems. Ependymal cells are cuboidal epithelial cells that line the internal cavities or ventricles of the brain and the central canal of the spinal cord. These cells have slender processes that branch extensively to make contact with other glial cells in the surrounding nervous tissue. Ependymal cells and nearby blood capillaries together form a network called the choroid plexus. The choroid plexus produces cerebrospinal fluid, a clear liquid that bathes the CNS and fills its internal cavities. The ependymal cells have cilia on their apical surfaces that help circulate the cerebrospinal fluid. Lastly, oligodendrocytes. These are large cells with a bulbous body and slender cytoplasmic extensions or processes. The processes of oligodendrocytes ensheath portions of many different axons, each repeatedly wrapping around part of an axon like electrical tape wrapped around a wire. This protective covering around the axon is called a myelin sheath. The supportive cells or glial cells of the peripheral nervous system include the satellite cells and neurolemmacytes, also referred to as Schwann cells. Satellite cells are flattened cells arranged around neuronal cell bodies in ganglia. Recall that a ganglion is a collection of neuronal cell bodies located outside the CNS. For example, we can see how satellite cells around the cell bodies of sensory neurons located in a specific type of ganglion, shown here in this picture, and this ganglion is part of the posterior root ganglion. Satellite cells physically separate cell bodies in a ganglion from their surrounding interstitial fluid and regulate the continuous exchange of nutrients and waste products between neurons and their environment. Neurolemmacytes are associated with peripheral nervous system axons. These cells are responsible for myelinating these axons, a process to be discussed in the next section. So neurolemmacytes are very similar to the oligodendrocytes I just talked about. Similar, but not identical. So what is the process of myelination in the central nervous system versus the peripheral nervous system? Well, if we look at this picture, we can first see the myelination of the peripheral nervous system shown in these series of pictures. We see a neurolemmacyte starts to encircle an axon, much as if you were wrapping a piece of tape around your pencil. As the neurolemmacyte continues to wrap around the axon, its cytoplasm and nucleus are squeezed to the periphery. Actually, I prefer the analogy of taking a tube of toothpaste that's halfway empty and you push all the contents of the toothpaste to the neck of the tube, and then you curl up the flattened portion of the tube. So all the toothpaste squished over to the neck of the tube, to me, represents the cytoplasm, the nucleus of the neurolemmacyte being squished out to the periphery. Then the overlapping inner layers of the plasma membrane, that's the tube part, they're going to form the myelin sheath. Sometimes the name neurolemma is used to describe this delicate, thin outer membrane of the neurolemmacyte. In the central nervous system, an oligodendrocyte can myelinate a portion of many axons, not just one. And if you look in your textbook on the next page, I'm going to show you here, in the top picture, we see the oligodendrocytes. And a single oligodendrocyte can actually extend portions of these cytoplasmic extensions, and each of those would wrap extensively around a portion of a neighboring axon. So you can have an oligodendrocyte actually participate in myelinating multiple different axons as shown in this picture. So a neurolemmacyte, an entire neurolemmacyte, myelinates a portion of an axon, but an oligodendrocyte can send out multiple extensions and provide myelination many different times on a single axon, as well as provide many different times for myelination on other axons as well. So the two pictures of myelination for the central nervous system and PNS are shown here side by side. Now when it comes to unmyelinated neurons, they still have the neurolemmacyte, they still have that there to kind of help and support the axon. The difference is that it doesn't wrap around the axon over and over and over again. Instead, it's just sort of like the axons are loosely stuck in to the neurolemmacyte cell membrane, as shown here in the right picture. When it comes to damage to nerves, students often ask, can they regrow? Well, let's explore that. It's true that the peripheral nervous system, your nerves, the axons, are vulnerable to cuts, crushing injuries, and other types of trauma. However, a damaged axon can regenerate if the cell body remains intact and a critical amount of neurolemma remains. The success of the PNS axon regeneration depends upon two factors. 1, the amount of damage, and 2, the distance between the site of the damaged axon and the structure it innervates or terminates on. The possibility of repair is decreased with an increase in either of these two factors. That means if the damage is extensive and the distance between the damaged axon and the structure it innervates is too great, then it's not very likely that the regeneration will occur. Neurolemmacytes play an active role in regeneration, and this process is shown here in the following steps. 1, the axon is severed by some type of trauma, so we see that in step 1. Step 2, the portion proximal to the trauma seals off by membrane fusion and swells. The swelling is a result of axoplasmic flow from the neuron cell body, and remember, axoplasmic simply means the cytoplasm in the axon. So we get some cytoplasmic flow from the neuronal cell body down to through the axon. And at the same time, the distal part of the axon that was severed from the cell body and the myelin sheath surrounding it breaks down. And macrophages are going to help remove the debris. In step 3, we see that the neurolemma, in conjunction with the remaining endoneurium, forms a regeneration tube. So remember, the neurolemma are the neurolemmacytes, and they're forming a tube. And then in step 4, we're going to see that the axon regenerates and remyelination occurs. The regeneration tube guides the axon sprout as it begins to grow rapidly through the regeneration tube at a rate of about 2 to 5 millimeters a day. That's actually pretty good. And this occurs under the influence of nerve growth factors raised by the neurolemmacytes. And then finally, innervation is restored as the axon reestablishes contact with its original structure. The structure is either a receptor for sensory neurons or an effector for motor neurons. Potential regeneration of damaged neurons within the central nervous system is very limited for several reasons. First, oligodendrocytes do not release a nerve growth factor, and in fact, they actively inhibit axon growth by producing and secreting several growth inhibitory molecules. Second, the large number of axons crowded within the CNS tends to complicate regrowth activities. Finally, both astrocytes and connective tissue coverings may form some scar tissue that obstructs axon growth. Now let's talk about some coverings of nerves. Remember, nerves are a collection of axons in the peripheral nervous system. And these words that I'm going to talk about have a very similar sound and prefix and organization to the F words that I told you about for skeletal muscle. Remember, fascicles and epimysium and that sort of thing. So we're going to have similar words for wrappings around axons or a collection of axons. So a nerve, like I said, is a cable-like bundle of axons. And an individual axon in a myelinated neuron is surrounded by neurolemmacytes and then wrapped in the endoneurium, a delicate layer of areolar connective tissue that separates and electrically isolates each axon. A group of axons are wrapped into separate bundles called fascicles, and they also are wrapped with a cellular, dense, irregular connective tissue layer called the perineurium. And this layer supports blood vessels supplying the capillaries within the endoneurium. All of the fascicles are bundled together by a superficial connective tissue covering termed the epineurium. This thick layer of dense irregular connective tissue encloses the entire nerve, providing both support and protection to the fascicles within the layer. This is a very important concept, the idea of neural communication, or the forming of a synapse. Synapses are intercellular junctions where two excitable cells come in contact to exchange information. A synapse occurs where the plasma membrane of a presynaptic neuron, synaptic knob, comes in close proximity to the plasma membrane of a postsynaptic neuron. So if we see here, we have two neurons in picture A, upstream neuron, downstream neuron. So the upstream neuron is called the presynaptic neuron, and its terminal knobs or terminal boutons would be considered the presynaptic membrane. And then the dendrites of the postsynaptic neuron would be considered the postsynaptic membrane. And down in the picture B, they're trying to show you with a cartoon kind of look. I think of these as dead stick figures myself. They're trying to simplify one neuron communicating to the downstream neuron. Again, presynaptic versus postsynaptic. There are different kinds of synapses. That means there are different places of potential communication between a presynaptic neuron and its downstream target. It's not always true that you have the presynaptic neuron communicating with the dendrites of the downstream neuron, as shown in the axodendritic synapse. Sometimes the presynaptic neuron can synapse directly on the soma of the downstream or postsynaptic neuron. That would be an axosomatic synapse. And then, to complicate things even more, we can have synapses occur between the terminal boutons of two different neurons. So that's an axo-axonic synapse. And for testing purposes, I'm not going to consider the axo-axonic synapse. I just want you to know that these kinds of functional contacts do exist. Alright, the synaptic communication, as we learned in our skeletal muscle lecture, means that a presynaptic neuron, when the action potential reaches the terminal bouton, we have synaptic vesicles that fuse and exocytose and release neurotransmitter on the next downstream neuron. And the neurotransmitter can bind to ligand-gated channels, and this can change the membrane potential of the next downstream cell. And as we learned, if the depolarization is enough, such that it opens another kind of channels, remember voltage-gated channels, then voltage-gated channels open because of this change in the membrane potential, and they open in an all or none fashion. So in this picture, you're seeing a resting membrane potential. We see that the inside of the cell is more negatively charged than the outside, and you should be able to tell me 3 major reasons why the inside of the cell is more negative than the outside. In part B, we see that if the cell depolarizes, that means gets less negative inside, more positive. If it does that enough and it opens voltage-gated channels, then they all open, all or none, all the way down the axon. This is called a nerve impulse or an action potential. And remember, that's the sodium voltage-gated channels opening. Which channel will open next as a result of the cell depolarizing even more? Potassium voltage-gated channels. And when they open, potassium leaves. And this helps restore the negativity inside the cell membrane. So again, the non-action potential can get down to the terminal bouton, and calcium voltage-gated channels open, as shown on the right picture here, and that leads to synaptic vesicles fusing and exocytosing. And again, this can keep continuing from one neuron to the next, or even from a neuron to a skeletal muscle cell, or some other involuntary effector. And in the right picture, you're seeing that there are ligand-gated acetylcholine receptors here, where the acetylcholine binds to them, and when that channel opens, we see sodium flowing into the cell. We now know that sodium brings with it a positive charge, and that means the inside of the cell becomes less negative, more positive. There are other kinds of synapses besides this electrochemical synapse. In the left picture, you're seeing 2 excitable cells inserting gap junctions. That, remember, we talked about this being inside a hotel room, and inside your room you have another door that if you open that and the neighbors next door in their hotel room open their internal door, then you can walk from room to room without going out in the hallway. So with gap junctions, each neuron needs to insert their own channel through their cell membrane and align these so that you have straight communication from one cytoplasm to the next. And we see this a lot in smooth muscle. They form these gap junctions, and this allows the electrical current to flow from one cell to the next. And in these previous pictures, the textbook shows that it's one neuron communicating to the downstream neuron in a linear fashion. And it's not really like that in our brain or spinal cord. We can have multiple upstream neurons communicating to a single downstream neuron. That's called a converging circuit. Similarly, we can have many upstream neurons communicating to many downstream neurons, such that there's more communication to downstream neurons. That's a diverging current. So we see one neuron communicating to 3, and 3 neurons communicating to 5, and 5 potentially communicating to 10, so that's diverging. We even can have circuits of neurons where an upstream neuron talks to a downstream neuron, talks to another downstream neuron, talks to a fourth neuron, and then the information can loop back up again. And then there's this quirky one called parallel after discharge, and it basically means that an upstream neuron can diverge and communicate to 2 different sets of neurons with their own communications, and then ultimately these 2 streams of neurons can diverge. So parallel after discharge simply means you diverge and then converge all over again. Alright, that concludes our introduction to our nervous system. On to bigger material as we talk about the parts of the brain and what their functions are, as well as learn about special senses.